I see quite a few familiar faces from last evening and the last session. But for new people in the room, I know everyone's got a slightly different schedule. I'm Larry Jameson, the Dean at the medical school. Welcome back for Alumni Weekend. We had a terrific scholarship reception last evening, a lot of white coats in the room, appreciative students for their scholarship support, and we just finished a panel with 50-year alums. It was super interesting. I know some of you heard the incredibly varied and compelling life stories and career paths and questions about, what might I have done differently and how my life have turned out. But from my perspective, that group couldn't have done it any better. And it's a natural transition from the last 50 years to hear about some of the things that are happening right now at Penn Medicine, at the forefront of medicine. We've got a terrific panel that John Epstein will introduce to you in a moment. But this is a very experienced group presenting and describing some of the things they're working on. And so I can guarantee you're going to find this incredibly interesting. And I'm going to turn the discussion over to John Epstein. John Epstein is my partner in the medical school, serving as the Chief Scientific Officer. And John himself could readily be, on this panel telling a terrific story. His background is in cardiology and developmental biology. His laboratory has studied the genetic and signaling pathways that regulate cardiac development that has a lot of implications for congenital heart disease and heart failure. He publishes in high-impact journals consistently, and including currently while he's carrying on these leadership roles at Penn Medicine. As you might expect, he's been recognized by a number of awards and elected societies. He's a member of the American Academy of Arts and Sciences, the Institute of Medicine, the National Academy of Medicine as it's now known. He's a member of the Association of American Physicians, and he served as the president of the American Society of Clinical Investigation, and really is doing a terrific job of helping us think about the future of research here at Penn Medicine. So John, let me welcome you up here to guide us through the discussion with this terrific panel.
Thanks very much, Larry. It's a real pleasure to be here, and it's been a real pleasure and a lot of fun working with Larry over the last couple of years as we do our best to support people like this. We have an excellent panel tonight of just a selection of some of the really world-renowned faculty that we're lucky enough to be able to work with and call our colleagues here at Penn. Before I get to them, I'll just make a few very brief comments. I've been at Penn about 20 years now. I came here as an assistant professor in the Department of Medicine. Was lucky enough to be asked to chair one of the basic science departments and now to work closely with Larry as the chief scientific officer. It's been quite a ride at Penn, and I hope your visit here this weekend, and many of you I can look around the room and see are quite connected with us. I think Penn is the best medical school in the country right now. And we have the tremendous advantage of being a truly unified health system. And the way academic medicine, research, clinical care has evolved, it's now very hard to draw a sharp line between what's medical school and basic research and what's clinical care and the hospital. And I think some of the greatest new advances are taking place right at that interface, where we're doing first-in-human trials, where we're moving ideas that used to seem miles away from ever really being a treatment in patients. Now we're seeing it in real time happening. And I honestly never thought I'd see that in my career to the extent that we're seeing it now. And I know you've all heard about the advances in immunotherapy for cancer, gene therapy after a rocky history now coming to fruition and curing blindness in kids, the hope of lots of other cures to come. And that in the background of these incredible advances that have been made, around the world in genome editing and our understanding of immunology generally, it's a pretty darn exciting time to be in this business. And as I travel around the country and visit our colleagues at many of the other great universities, I think Larry has the same impression. We always come home and say, boy, I'm glad I'm at Penn. And it's a very nice feeling. I think you'll sense that as you talk to faculty members, students. Boy, I think it'd be a great place to be a medical student right now. We have the largest MD-PhD program in the country. I think it's the best MD-PhD program in the country, and I think that most people around the country would say that. A number of other areas, especially at the interface of clinical care and research. So I think we're all pretty proud to be here today and to try to show off some of the latest discoveries and to have a conversation with you about what the future holds. So I'll just briefly introduce our panelists. We're going to give them a couple of minutes to present a few of their ideas to set the stage, and then we'll open it up for questions. And we'll go wherever you lead us in terms of the questions that we'll discuss. So I'll start with our last speaker. I'll go in reverse order with my introductions. And Scott Levin, raise your hand, Scott, is the Paul Magnuson Professor of Bone and Joint Surgery. He's chairman of the Department of Orthopedic Surgery. He has the unusual training in both orthopedics and in plastic surgery and in about 5 other subspecialty areas as well. He's really pioneered microsurgical techniques, and many of you have probably seen him on TV or read about the truly impossible types of surgeries made possible by his work, most notably perhaps the hand and arm transplants that have happened, but other cutting-edge areas he's likely to tell us about. He got his medical degree from Temple, his residency in orthopedic surgery at Duke, and he trained in various fellowships in pediatric orthopedics, hand, microvascular surgery at the Shriners Hospital in Louisville, Kentucky, and also at Duke. He's published over 250 articles, something like 70 book chapters, and maybe more importantly, he's a pillar of the institution here at Penn. He's a wonderful colleague who I enjoy working with tremendously. He's the doctor's doctor. And when you have to call him for an emergency, as I've had to do on too many occasions, you don't have to wait 5 seconds for Scott to get back to you with a caring tone in his voice. He's the kind of doctor you want. Virginia Lee, known to all of you, I think. John Ware Professor in Alzheimer's Research in the Department of Pathology and Laboratory Medicine. What I didn't know until fairly recently is that she actually studied music originally in the Royal Academy of Music in London before she went on to get her various other degrees, including a PhD from UCSF. She's worked in the private sector. She has an MBA. She joined the faculty here in 1981, been a long-standing member of the faculty who's mentored generations of outstanding trainees now running their own labs around the country. Working with her husband, John, they're amongst the most widely cited, best known, and most productive researchers in the field of neurodegeneration. And they've, over the years, made a series of seminal discoveries that have continued to move the field forward, especially about the proteins that can aggregate in diseased neurons and how those aggregates actually lead to toxicity in those neurons. She's won all sorts of awards. I won't list them all, but I'll just mention the Lifetime Achievement Award from the Alzheimer's Foundation, the Passero Foundation Award for Neuropsychiatry Research, and most recently the John Scott Award from the City of Philadelphia for inventions that have contributed in an outstanding way to the comfort, welfare, and happiness of mankind. That's pretty good, I think. She's a member of the National Academy of Medicine, she's the Board of Scientific Counselors to the National Institute of Aging, truly an international expert in Alzheimer's, Parkinson's, those sorts of disorders. Michal Elovitz is professor of Obstetrics and Gynecology, vice chair for Translational Research at Penn. She's also the director of Maternal and Child Health Research Center, director of the Maternal Fetal Medicine Fellowship Program, director of, I'm sure, some other things as well. She's a prolific physician scientist, really a role model for our trainees. And she's an international expert on the causes and therapies for preterm births. And she was instrumental in fairly recently bringing a large March of Dimes grant to Penn for a center that focuses on prematurity research. She's published in a wide spectrum of areas, from the role of microRNAs that are made in the placenta and can contribute to preterm problems, to eclampsia, to other complications of pregnancy, the use of progesterone and the settings of preterm labor, and she'll talk to us about some of the latest discoveries. It's actually a fascinating area of research these days. And finally, our first speaker, and I'll, by way of introduction for his comments, Kojo Elenitoba-Johnson is the director of our Center for Personalized Diagnostics, and he's the Peter Nowell Professor of Pathology and Laboratory Medicine. He's an expert in this emerging field we all hear about of precision medicine, and especially as it relates to cancer, genetics, and targeted therapies. And in particular, he not only is an expert in the genomics and next-generation sequencing that characterize that field, but also the proteomics, mass spec approaches to understanding post-translational modifications of proteins in the serum that can characterize certain disorders and in tumor tissues. Kojo has a remarkable life story and is a wonderful colleague here at Penn. He comes from Lagos, Nigeria originally, and as the story goes, Kojo, you correct me if I've got it wrong, but it wasn't so easy to find a medical school to come for his residency in this country. He got a spot at Brown. Some people wondered whether this guy could, would do okay, and he ended up at the top of the pack and chosen as the chief resident there, and has gone on to a stellar career after that. Proving to all of us who's who around here. I very much enjoy working with him, and I look forward to his comments to start us off.
Thank you very much for that generous introduction. The person I would most like to have heard that is my mother, who I would suspect so. She sort of goes everywhere with me and asks me questions that I wouldn't have anticipated she would have been attentive to. It's a great privilege for me to get a chance to speak with you all. So I'd like to recognize my co-moderators, of course, Dean Jameson. Someday when you all have 6 hours to kill, I can tell you how I got here, but that by itself is more entertaining than it is serious. So today what I was going to try and do in the 8 minutes that I was given, and I have my eye on the clock over there, is to give you an idea of the transitions we've had to go through in the field of cancer that would have substantial relevance to all other disorders that are linked to diseases of chronicity and some acute diseases. So the foundational basis for precision oncology. So everywhere you go, on the radio, on TV, you hear about this entity or that entity practicing precision medicine and so on and so forth. The foundational basis for all of that started here. And that started with the discovery of the Philadelphia chromosome, which you all know was discovered by Peter Nowell and David Hungerford. I have what I consider to be an awesome privilege to be the inaugural Peter Nowell Professor. And it's certainly something I didn't dream of walking on the streets of Lagos, Nigeria, when my mom was trying to tell me that I could do better. By way of history, I just wanted to give you a sense of how important this observation was. So as early as the 1890s, there was observations that there were mitotic aberrations in tumor cells, and that was it. So imagine how much time had to elapse before a seminal observation actually confirmed that cancer is a genetic disease. The vast majority of cancers are acquired genetic diseases, and there's several important implications that arise from that, which I will touch on. Now, I want to underscore that at this time, so the Philadelphia chromosome was found in connection with chronic myelogenous leukemia, or CML. At this time, even 5 years prior to the observation, it wasn't settled how many chromosomes we had as humans. You know, it was varying between 48 and 50. And last I checked, my chromosomes, they were 46. I have a Y chromosome. I have to apologize for its diminutive size, but both my wife and my mother are very forgiving with those lapses. It took another 13 years before Janet Rowley at the University of Chicago found another translocation. So even in the time at which this original discovery was made, there was still a tremendous amount of skepticism in basically establishing that was this an epiphenomenon or was it linked at all to causality. Now this is really critical to keep in mind. So we go through a number of transitions and ultimately it starts getting incorporated into the clinical realm over a period of more than 28 years. And if you go right back to David von Huntsman's observation, we're talking about a gigantic leap for us to have gotten here. And of course, Brian Drucker was able to work with Pfizer to come up with a drug that made this the first and the best example of precision medicine, a precision drug targeting the specific mutations occurring in cancer. This is huge. So just again for context, cancer is a leading cause of death globally, 14 million new cases annually and more than 10 times that number living with cancer. And today, 1 in 6 deaths is due to cancer. So I'll just shift a little bit and talk about the power of DNA, since that is a significant arm of what my daily activities is. So we have 3 billion letters, all of us, again in men, give or take a little subtraction, but it's okay, we're still functional. With 3 billion bases, basically enumerates your code of every cell. And with only a few markers, we can type everybody who's on the planet currently, that's 7.2 billion people estimated. We can type everybody who has been on the planet prior to now, and we can type everybody who will ever be on the planet, okay? So to me, that's what I call muscle. DNA is the ultimate digital language, so it's interfacing with the current technology computationally is a natural transition. And this, obviously, as you can tell, reflects the diversity of all humans. So what if we had the ability to harness interrogation on a genome-wide scale into cancer cells the way we can do it, the way I described, we could type every human being born and yet unborn. It has critical implications that flow from the discovery of the Philadelphia chromosome, which I said is an acquired abnormality. So the vast majority of cancers are acquired genetic disorders. So this brings me here where I am currently the director of the Center for Personalized Diagnostics, where we use these technologies to basically interrogate cancer. And we do this for 5 reasons I'll tell you in a minute. I'll tell you that since being here, our numbers continue to climb, and we're anticipating that at the end of this year, we would have tested 11,000 patients in excess of 11,000 patients who have cancer using these sophisticated genomic techniques that make it possible for us to interrogate mutations rather than one at a time. We can interrogate the vast majority of disease-causing alterations. So one of the inescapable consequences of the Philadelphia chromosome, the birth of cancer genetics, is that we can now make confident and correct diagnoses rather than have opinion that can't be verified other than by me pitting my ego against that of another well-trained individual, and that doesn't help any patients. So I show this image, and I apologize for its graphic nature. This is Burkitt lymphoma. The physicians in the audience will be well familiar with. It's common in Africa in what's called the malarial belt. I make that point to help people remember that not until 1960 did we know what the cause of cancer was. I have a side story. Malaria is blamed for everything in Africa. In my case, it was blamed for growth retardation. Consequently, I feel rather cheated. I could. Had an alternative career funding my research with my compensation from the NBA. Malaria is blamed for mental retardation. And I find myself here amongst, the smartest people in the world. And I'll also tell you that I have had malaria more than 200 times. So imagine what I could have been if I didn't have malaria. What I'm trying to show you here is that this jaw tumor, which is characteristic, is the same tumor as the one in this patient's abdomen. This tumor has the fastest doubling time known to man, and what this is supposed to illustrate is that the location of the tumor does not tell you what the tumor is. So if individual A says they have breast cancer, and individual B says they have colon cancer, there are some with identical genetic abnormalities that gives us the opportunity to start thinking about a molecular taxonomy rather than disease by anatomic site. So that's a profound change in the way we've thought about things, and I'll show you an example of that. I have to say that the alteration is characterized by this genetic abnormality associated with MYC, and one of the world's experts in the metabolic consequences of MYC activation is Chi Dang, who's here. The second consequence is that we can monitor disease before we see it physically. We can monitor disease by the molecular abnormality at a very high sensitivity, 1 out of 1,000,000 cells, and as the technology develops, even better. The third consequence is targeted therapy, which I talked about. Imatinib, which was used to target the consequence of the Philadelphia chromosome, is well known to you all. And you know you've really made it when your discovery makes the front cover of Time magazine. Forget about the high-impact journals that John Epstein publishes in. I show you another jaw tumor basically to say tumors of the same location may have dramatically different genetic profiles. And this is one that arose from work in my lab where we discovered this red jaw tumor, which is a disfiguring tumor for which there is no real cure, surgically excised, characterized by recurrences which lead to disfiguring abnormalities even in kids, as in this example. And you can see here the genomic profile of this has a number of genes. A good example of this is BRAF, to which a drug, vemurafenib, exists and specifically targets this jaw tumor, obviating the requirement for surgery, even in metastatic cases such as this. Again, one personal story, right after publication of our paper, an individual had metastatic ameloblastoma, shown by these red spots here in the jaw and the lung, got treated and was massively responsive. I can't talk about precision medicine in this part of the universe without recognizing the work of Carl June. And the fifth consequence of recognizing that tumors have acquired mutations is that you could actually target those using the immune system. And that is going to be a dramatic consequence of that observation. And we will get to the point where personalized cancer vaccines are being deployed in the armamentarium against cancer. Again, you know you've made it when in 1960 you make an observation, but it comes to the awareness of the presidency 50 or 60 years later. And everybody is trying to be associated with this success story. So I just terminate by saying this is the world leader in precision medicine without any question. And it is a massive privilege to be part of this company. Thank you very much.
The next speaker will be Michal A.
They should get out.
There's a hand clicker also.
Okay.
It's a pleasure being here. I have to say, Dr. Jamieson and Dr. Epstein talked about what makes Penn different. I feel so incredibly short right now. I need a stool. I was not growth restricted, and I'm still vertically challenged. I would challenge you to ask your colleagues in medicine when they go to their reunions or when they go back to their schools, is there an OBGYN sitting on a panel about research? I would venture to tell you there's not. So while I think you would expect to hear about precision medicine and oncology and transplants and cardiovascular disease when you came to Penn, I'm not sure you expected to hear about reproductive health. This is what makes Penn so unique and such a pleasure and exciting place to work, is because as a maternal fetal medicine doc, I'm gonna tell you about things that are happening in the world of obstetrics that are hopefully gonna have a tremendous impact on the lives of women and children, not just here, but globally. So the title, as you can see, of my talk is Preventing Preterm Birth, and it's time. So for those of you in the audience who maybe are not so familiar with prematurity, I'll just give you the numbers. $26 billion a year in the United States. That's what it cost us. Not for lifetime, just total for a preterm birth. Globally, every year, so I think you just saw a number of 14 million. I'm a little competitive. 15 million babies globally are born preterm. 15 million. When the United States, while we lack in ratings for perinatal survival than we should, we're still talking that preterm babies here live. There are many countries where preterm babies born at 30, 28 weeks, or even 32 weeks die. So this is not oh, I just wish they were born later and were a little bigger and a little taller. This is having babies that will live differently for their entire life if they survive. You'll notice on this slide, maybe not, that the preterm birth rate is very disparate. So it's about 12% in the United States, but in non-Hispanic Blacks, it is 16%. In this city, where you're sitting right now, it's 18%. Think about it, that's almost 1 in 5 African-American women will give birth to a preterm baby. That is insane, and we don't understand it, despite decades of doing this. What I really want to drive home, though, it's not just that you're born early. If you were born early and you had no adverse outcome, who would care? What you must understand is that ex-preterm children are at high risk for a spectrum of adverse neurobehavioral disorders, from cognitive dysfunction to cerebral palsy to autism. I had a patient, I run our prematurity clinic here. I direct that too. I run the prematurity clinic. I had a patient sitting across from me who had a 24-weeker who passed away. She wanted to know what it meant if the baby had lived. She was having a hard time getting past it. So at our institution, our neonatologists are amazing. Our survival rates at 24 weeks are 50 to 60%. There's one problem. Of those that survive, 80% have major neurological disability. And I think for most of us in the room, when we say major, it's not what we would, that major would seem life-threatening. If you think about all of them will have moderate disability just means you're blind or deaf. That you only have cerebral palsy that affects 2, not 4 limbs. That your IQ is only 2, not 3 standard deviations. So this is a lifetime disease. These children have decreased lifetime expectancies and long-term medical problems. So how do we address preterm birth? I just gave, I believe, a pretty convincing argument we need to do something about it. So what do we believe? How does this happen? So this is a schematic from a paper that I wrote 13 years ago, and this is what we believed until a few years ago. Bacteria colonize the cervical vaginal space. They go up through the cervix, and shockingly, despite what most of you probably know about immunology, we thought this had no effect. They just kind of floated up. They replicated in what's called the decidua in the uterus, in the placenta, and then crossed to the fetus, either by the umbilical cord or breaking through the fetal membranes. That colonization of bacteria elicited immune response, led to uterine contractions, which in turn caused cervical remodeling. Seems to make perfect sense, yes? So for decades, this was the paradigm. So what did we do as doctors who need to fix things? We did all these clinical trials targeting myometrial activity. If we stopped uterine contractions, we could prevent preterm birth. We spent billions of dollars on randomized clinical trials looking at all different ways to monitor and prevent preterm birth. And you know what happened? Not a thing. Didn't change the preterm birth rate. So then we focused on uterine infection. Surely if we stopped those bugs from flying up into the uterus, we would prevent preterm birth. So again, we spent a lot of money looking at different trials. And what we found after the last 40 years, no benefit, no change in the preterm birth rate. So I'm going to talk about an organ you may not be so familiar with, or maybe you should be. It's called the cervix. The cervix does this very amazing thing that we have ignored for the last, I don't know, few hundred years or so. When you start a pregnancy, your cervix looks something like my hand, like this. It's closed, it's served to protect the mother from ascending infection, and it stays that way for the most of pregnancy. And then lo and behold, by some miraculous thing that we still don't know that happens, somewhere between 37 and 40 weeks, that cervix opens slowly. And then what happens to the cervix? I know there's at least one of you in the room. It opens to allow us maybe a small head or a big head to come through, but basically to deliver between a 6 and an 8-pound baby. And if you remember your OB rotation, you might remember that when you went to go feel, to make sure it was a head and not a tush coming down at you, you actually can't feel the cervix. The cervix disappears. There's nothing there. If anyone can name another organ in the body for me where it comes solid tissue and then disappears, raise your hand. There's not one. There's no other tissue that does that. Okay, so it's a malleable tissue. We all know about collagen and other things can change. So that must mean it would take weeks or months for it to come back, right? I mean, it shouldn't just reappear right after the baby comes out, but it does. Because if it doesn't, the woman dies. If that cervix stays open, she bleeds to death. So not an hour, not 5 hours, not even 15 minutes. 3 to 4 minutes after that delivery, that cervix is back to what it was, if not tighter, thicker, and we don't understand this. And by not understanding how the cervix works as both a connective tissue organ, as a epithelial mucosal barrier, as an as a bridge to the immune response, we are not going to understand preterm birth. So we decided this is where we should start. So the other great thing about Penn is when you start saying, I don't understand. What about reproductive tissues do I not get? It's a mucosal barrier, it's an epithelial barrier, it's connective tissue. What do? Well, at Penn, you go start bothering people. You knock on a lot of doors. You find your favorite immunologists, you find your favorite orthopedics who does bioengineering and collagen work, you find your microbiologist, and you start to rethink from the start. And here you get to do this. So this on your left are epithelial cervical cells. Similar to the gut, and this is how we kind of mimic this paradigm, there are bacteria sitting on it. Those little green and red things are different immune mediators that are normally present in an epithelial mucosal barrier. What we proposed has happened is that the bacteria, these very common bacteria that live in the cervical vaginal space, change. And when they are dysbiotic or abnormal, they create a bad immune response or an unfavorable. And I'm not going to characterize it because I don't think we know enough about it, but it changes and then it breaks down the barrier to the cervix, what leads to premature cervical remodeling and then uterine contractility and preterm birth. So we've changed the paradigm to say it's the cervix first. So how have we really approached this? Well, in a multitude of ways that with 8 minutes I don't have the time to tell you, but I can tell you we have focused on the microbiota, the metabolomics, the molecular expression of what's happening there, the biomechanics, which despite what I just told you about the cervix, has never even been thought about, and the immune response that occurs in this space. I'm going to quickly just show you a result of a R01-funded 2,000-person cohort in women, pregnant women. And what we did is we asked women to self-sample during 3 points in pregnancy, and we looked at the outcome of preterm birth or term delivery. And out of those 2,000 women, we had about 120 cases of preterm birth, and we compared it to 600 controls. This is a heat map. This first group you'll see are the different time periods in pregnancy, which were all before 28 weeks, so early in pregnancy, in white women. The red means that those bacteria were strongly associated with preterm birth. The blue, catch this, means they were negatively associated. They were protective against preterm birth. So you can see, I'm showing you 1 out of 250 species. We found really big magnitude associations between bacteria and preterm birth. Remember what I said about the disparity in races and preterm birth? This is the heat map of African American women. So still bugs really strongly associated in those that are protective, but very different than Caucasian women. So I want to give you an example of what we've done because I think it's so impressive and biologically interesting. One of our good bugs, my chair loves how I call them good and bad bugs, by the way. Not really, but we'll pretend she does. This is Lactobacillus rhamnosus. It's a lactobacillus. It should be good. This one is. But what we've done, very different than what's been done in other worlds in the microbiome, is we've done the absolute abundance. How much bacteria do you have? So on your x-axis is bacterial load. On your y-axis is preterm birth. With Lactobacillus rhamnosus, when you have a lot of bug, your rate of preterm birth is 0. So if you have enough good bug, you don't have preterm birth. Mobiluncus, my lab loves this bug. They think it's a funny name. Mobiluncus. You have high rates of high load of bacteria, and your preterm birth rate is 50%. Good bugs, bad bugs. Going to change the way we treat preterm birth. So how do we move forward and make a difference in this field? So this is a very active field of research for us. I'm not going to spend your time telling you, but I can tell you with each of these items I've talked about, we have made forward movement in understanding what's happening. I don't have time to talk about it today, but I just want to bring in, because I'm sitting next to Scott, and someone made the comment, he looked at me and goes, so Lou's on your team? Lou Soslowski, who's, again, one of our associate deans here at Penn, who's a bioengineering guy, works on tendons. Guess what? He's on our grant. We have him working on the biomechanics of the cervix. I challenge you to find in other med school where a bioengineer and a maternal fetal medicine sit down at a table and talk about the cervix and what it does. And this, along with the microbiology work that we're doing, I really think is going to lead us to this. Our goal. So as a scientist in the beginning of my career, the goal was just to figure things out, to learn things, to know things. I'm getting older. I want to make a difference, and that is what we're trying to do. So what we believe we can do is determine who is at risk and then to offer new therapeutic strategies. We think we've actually reached that point with our new data that I did not show you all, that we are actually going to be able to profile women, and I mean the 4 million pregnant women in the US and the million of women worldwide, to say who is actually at risk. So similar about the first talk, to really give a personalized precision medicine approach. So we're going to do that, and then what we think has to happen in tandem is that we need more tools to assist in identifying. And our goal is to do that by biomechanics, using the Sing Center here at Penn, yet another great thing at Penn. We are intending to use biosensors, pull the two together to figure out who's at risk, adequately treat them based on the risk, and prevent preterm birth. And I think that's the end of my slides. Hope that wasn't over 8 minutes. Thank you.
Virginia Lee will be our next speaker, and today proves the adage that you can never
find
use
it?
academics to their 8-minute limits.
Yes,
So
great. Yeah.
we'll try to leave a few minutes for questions.
Okay.
You have to point it at the screen, this screen.
What? I can do, okay, yes. Okay, and during my introduction, and John said that I've been here for a long time. In fact, I have spent my entire career, research career, at the University of Pennsylvania. And the work that you're going to hear me talk about would not be possible, but not for the collaborative spirit in the School of Medicine. And so I think that, you know, and as you will see, that we've really accomplished a lot. So why are we so interested in Alzheimer's therapy now? You know that, let me just advance the slide. Shit. Okay.
Just point it this way.
Huh? I have to use this one. Yeah. So because of the fact that the global population forecasted Americans are getting grayer. And as you get grayer, your risk of getting Alzheimer's disease increase. And also that if you look at the demographic, and in terms of from the population point of view, that over the 100 years, and from about 1950 to 2050, and the birth rate is dropping. And so you see that the age of people under the age of 5 is decreasing, whereas the age of people over 65 are increased. So because of this change, that we are all at risk to develop an Alzheimer's disease and related dementia. And so, and many years ago now, 100 years ago, that it was shown that an Alzheimer's disease, and one of the major problems is the accumulation of these misfolded neurodegenerative disease protein in brains. You can see this lovely picture. And I actually took this from a foundation called QPSP. And the pink stuff in the brain is symbolizing the brain trash that accumulate as you age and when you develop Alzheimer's disease or other related neurodegenerative diseases. So we know about that, and when I started my career, so we wanted to do something about this. We want to understand what are these proteins they're accumulating in different neurodegenerative diseases. And so starting in the late 80s, and John Trojanowski and myself, and systematically and went after each of these disease proteins. They're misfolded in Alzheimer's, Parkinson, and also amyotrophic lateral sclerosis. And we identified tau protein and as the building block of neurofibrillary tangles in Alzheimer's disease brain. And we also identified, this was done in 1991, and in 1997, we identified alpha-synuclein as a building block of these inclusion called Lewy bodies in brains of Parkinson's disease patients. And then finally, in 2006, we discovered that this protein, RNA binding protein called TDP-483, is misfolded and accumulate in motor neurons of patients with amyotrophic sclerosis. And so with that, and what then you see this pathology, then what do they do to your brain? And basically, it causes brain degeneration. And so you see here in an extreme case of Alzheimer's disease, and you can see on the left side, on this side, the Alzheimer's brain, which is very, very much shrunken compared to the control brain. So suggesting that there's diffuse atrophy, neuron loss, and then with this disease progression, and eventually lead to loss of cognition. So in addition to neurofibrillary tangle in Alzheimer's brain, there's actually a lot of, there's another pathology, which is called senile plaques. And so both of these pathologies work in concert to cause Alzheimer's disease. And so, so when we know all of this information, then what do we do in terms of trying to develop therapy for Alzheimer's disease? So our approach was to take brain and like Alzheimer's brain like this. And then we basically, and because they have so much of these pathology in brain, we wind up the brain and then we generated Alzheimer's tau, so the pathological tau that I talked to you about. And then what we want to do is that we want to see whether we can develop a mouse model that showed the same pathology as in Alzheimer's brain. So we injected this material into the hippocampus. and the cortex of a regular wild-type mouse. And we wait for about 3 months, and we wait for different lengths of time. But at 3 months now, you can see that in the hippocampus, the mouse developed tangles. So what that means is that that's Alzheimer tau induced the endogenous mouse tau to form pathology that accumulate progressively over time. We also showed that they spread too in brain over time. So you can see here, for example, that we injected on this side, on the ipsilateral side, but you can see now at 3 months on the opposite side of the brain, the contralateral side, you can see accumulation also of neurofibrillary tangle as well. And you also look at regions that are distal, very far away from the hippocampus. And you see that in the locus coeruleus, for example, over time, there are more and more pathology. And that's what we think happened. And during the progression of Alzheimer's disease, the pathology spread to different parts of the brain and caused increasing deficit in the patient. And so this is a great a model for a neurofibrillary tangle. We can also actually do the same thing in cells. And why is this so important to do them in cells? Because then it'll give you a platform so that you can screen drugs to get rid of the pathology. So here, what we have is a primary neuron in culture. Again, we do the same thing. We took the Alzheimer tau, we just literally toss it into the culture and then wait for a certain period of time. And you see this green is all the tau pathology accumulating in these neurons. And at higher power, you can see that they have the same kind of rope-like structure as you would see in brains of Alzheimer patients. And so with this two experiment that I showed you, and it's really informative to tell us about the mechanism of what's going on in terms of the spreading of pathological tau in Alzheimer brain. So basically what you see here is that on this side is a sick cell which contain these misfolded tau. And basically what happened is that this misfolded tau get out of the cell, and if it wants to transmit, it has to get out of the cell, and then it needs to be picked up by a healthy neighboring cell. And once it gets inside, it corrupts the endogenous protein to adopt the bad conformation. And so over time, this keeps going on and on and on, so that's how it spread. But at the same time, it create also, understanding this mechanism, create opportunity for targeted therapy. So basically, we can have small molecule drugs that can block the release of the pathological tau, and we can also block the uptake of this bad tau into the healthy cells. And we can also prevent seeding of the endogenous recruitment of the endogenous protein to expand into more tangle-like protein. And we can also promote degradation of the material that's extracellular. And so what is the status now? With all that information, how do we use that information and for developing new therapy? And this slide basically summarizes what we know about an Alzheimer therapy today. And one most important fact that we know today that we didn't know 20 years ago is that Alzheimer's disease starts decades before symptoms. This is where you have symptoms. And so beta amyloid started accumulating in 10, 20 years before you have symptoms. And it increase. And then at the same time, a little bit more slowly, tangle develop as well. And so the two of them act together to cause disease. And so far, we have symptomatic therapy, as some of you may be familiar with symptomatic therapy. But now what we want to do is really to focus on modifying therapy. So, and tau-directed treatment is one such and modifying therapy, but there are others too. There's A-beta-directed treatment and also directed against inflammation and so on. So, but obviously, the ultimate goal, because the disease starts so early, is to be able to prevent the disease. So I think that there's just a lot going on, and I think that we're looking forward to, in our Center for Neurodegenerative Disease, developing tau-directed therapies. I thank you for your attention.
So our last speaker is Scott Levin.
Thanks, Jimmy.
While he's getting ready, I will say that transmissibility of tau protein, I found one of the most surprising results.
Fascinating.
And it really does open up whole
Somebody-
ways of thinking about
Somebody working at
Right.
Is somebody driving this or I'm driving it?
somebody's driving it.
Oh, well, that's better. So while my slides come up, I want to thank John Epstein and say good morning to you. I'm a simple carpenter and a plumber. My esteemed colleagues who you've heard from are the real geniuses of Penn Medicine. And my purpose over the next 8 minutes is to tell you a story. The definition of an orthopedic surgeon, the adage in your medical school class, strong as an ox and half as smart. But again, I'm very, very privileged to be here and going to talk to you about the topic, gifted hands. Let's go back in history. My mentor, who was David C. Sabiston Jr., who worked with Alfred Blaylock and went to the lab with Dr. Blaylock and Vivian Thomas and worked on the Blaylock-Talzig shunt as an intern and resident, emphasized the need for the surgeon scientist. And if we go back in history and the lessons of my mentor, Dave Sabiston, who was one of the greatest leaders in American surgery, we have to take surgery from the bench, or from the bedside to the bench, ask questions on rounds, go to the research laboratory, and come back to the bench with a solution. That's what Virginia and Michal and KJ have all shown you. We're asking questions on how. This is why we went to medical school. So one of my heroes is Sir Charles Bell, was a British surgeon, lived even before Ben Franklin's time. Hands are the ready instrument of the mind. What a profound statement in the time that he lived. So this is a story. Keep in mind, hands are the ready instrument of the mind of 3 patients. What generates advances? Clinical questions. So just imagine you're sitting here, you don't feel well. This afternoon or this evening, you or your loved one here in the audience winds up in the Penn ER. High fever, nausea, vomiting. Doc comes in, your physicians. You have sepsis. 2 days later on a ventilator, pressors. We can't sustain you unless we amputate all 4 of your limbs. So now imagine you're sitting here, checking your iPhone, hands crossed, you have no hands and no legs. That's the story of these 3 patients. Lindsay S., Zion Harvey, and Laura Natoff. So you have a loss of connection. Am I tugging at your heartstrings? Genuinely, my friends, I hope I am. Because this was a clinical research question. How do we give people who have no hands, hands? And Lindsay S., this young lady here, was a Virginia Commonwealth University graduate. She was in the fashion industry. She had Crohn's disease. She got sick and wound up like this, and came to see me several months after her calamity, knew I was interested in hand transplantation. In my mind at that time, it had been done in 1998 poorly in France. The first case had been done in this country in 1999 successfully in a man by the name of Matt Scott, who lost his hand in a road accident. Matt Scott sits on my UNOS, United Network for Organ Sharing, Vascularized Composite Allotransplantation Committee. 19 years later, he has a functioning transplanted hand from a deceased donor. And so less than 20 years ago, the field of vascularized composite allotransplant, the extension of solid organ transplant. Solid organ often is life-saving, cardiac, pancreas, lung. But what about the burned patient, the disfigured patient that has no face, that can't go out in public? What about the quadrimembranal amputee who has no limbs and who cannot don prosthetics for a variety of reasons? So I came to Penn in 2009. Let me remind you about our culture here. Let me remind you, one word, and I'm a relative newcomer to Penn. The word is collaboration. You heard about team science from KJ. You know, Michal working with Lou Kozlowski. Who would have thought that now I'm responsible for obstetrics and gynecology? It's orthopedics is 1-0, now I have OBGYN. And the brilliant work that John and Virginia do for promise for millions of Americans and people around the world. But let's come back to the core value of Penn and how did we take Lindsay S., who you just saw here, a life of despair, to doing CrossFit, working with our wounded warriors. How do we take little Zion Harvey, who at 2 years old lost his hands and his legs, and transplant him new arms, and our most recent patient who flew from Paris, France, from her transplant, was notified when she was in Marseille on vacation, flew to Paris, flew to Penn. 10 hours later, we gave her new hands. Let's come back to the essence of our mission here at Penn as a research institution. And this was an idea I had sort of after I met Lindsay. I was in an institution. Let me just give you the comparison. I was in an institution that didn't believe in making advances. I thought, well, if I go to the lab, Dr. Sabiston was no longer living, if I go to the lab and demonstrate that I'm doing benched research, asking the question of what if I see a patient who's already immunosuppressed and they need an abdominal wall, they need a face, they need a hand, I need the animal model. Dave Sabiston would never let me in the clinic proceed directly to clinical execution of vascularized composite allotransplant, which parenthetically, again, is the definition of using a deceased donor to provide body parts, face, abdominal wall, phallus, penis has been transferred to the MGH and in South Africa. We're on the verge of a new phase of allotransplantation and need to recognize pioneers, surgical pioneers like the late Joe Murray, who won the Nobel Prize in Medicine in 1999, awarded for his work in 1954, the Hurrick twins, kidney transplant, the entire field of transplant immunology. We're still struggling. All of us here have some sort of crossover. KJ, correctly, Virginia, correct, Michal, the issues of unlocking the mysteries of the immune system. And it's now in my world. We don't have a perfect solution to prevent rejection, but here was the experimental model, transplanting a heart into one rat, a second immunogenic challenge, an antigen challenge, to see if the patient who already had a solid organ transplant would tolerate a vascularized composite allotransplant. It did. So, other opportunities for research. The bottom line, we started the VCA program here when I arrived in 2009. We did our first patient in 2011, then Zion Harvey, if you will, the transplant, the first pediatric transplant of a vascularized hand in a child around the world. It was done at CHOP in collaboration and led by Penn. And when we transplanted Zion, here are more research questions. The neurologists were fascinated. Here's a child at the age of 2 who lost his hands. Remember the pre and post central gyrus? Virginia, that's the only neurology and neuroscience I know. But seriously, the advances in neural imaging have allowed us to look at the centers and the pathways in his brain that were non-existent. He lost his hands at 2, he had a transplant at 8. The brain went to sleep for 6 years. And now we're studying Zion and seeing how his brain is waking up, and he can feed himself, he can toilet himself, he can do all the things, as you saw, that Lindsay can do. So these patients are now on immunosuppression, but they've reconnected with their world, something that probably when you went to medical school and when I went to medical school, we never would have thought are possible. So John, I hope I've been under 8 minutes or just at about. And what's the future hold? Well, we have now the so-called orthoplastic limb salvage program at Penn, combining the principles and practices of orthopedic surgery, plastic surgery, vascular surgery, into advances in extremity reconstruction and salvage, as I just showed you. The absolute need for continued optimization of immunosuppression therapies, it's not going to come from me. It's going to come from the esteemed scientists sitting on the panel here. Hand transplant, face transplant, vascularized composite allotransplant falls under the category of regenerative medicine. Penn leads that globally. And finally, and I put this in for Michal, she wasn't expecting it. In addition to our successful hand transplant program, we're leading both in the pediatric and adult world. We've just embarked with Debbie Driscoll, the chairman of OBGYN, Christos Kalafaris, our director of infertility, and a promising, brilliant new attending surgeon, Kate McNeil, into the uterus transplant program. So talking about crossover, it's the UNTIL trial. Thousands and thousands of women around the world cannot have children because of anomalies in the uterus. Many states around our country, surrogacy is prohibited. And if you think I'm excited about hand and face transplant, the concept of a young woman who cannot have a baby, has a uterus transplant from a deceased donor, is on immunosuppression for a period of time, has 1 or 2 children, and then that uterus removed, is indeed Penn's promise for the future in this new field called vascularized composite allotransplant. I won't be delivering the baby, but we're collaborative, and the microvascular aspects of that, stay tuned, I'll be involved. So thank you very much for your time.
Well, if I can wipe the tears away, and every time that the story of Libby Zion gets me going. When I had the chance to meet him and see him running around and playing, and it really is something. We have time for a few questions, and I'll just open it up to the audience and game for anything. While you're thinking, Scott, give us a sense of the size of the team that's required for these sorts of hand transplants. Let's say one over at CHOP or something like that.
Well, I don't know if you can hear me. Well, science is a team sport, medicine's a team sport, reconstructive surgery is a team sport, and we have rehearsals in our human tissue lab that we started. The first transplant was 40 people, doctors, nurses, anesthesiologists, in and out of the operating room. We have 10 to 12 attending surgeons, all hand experts at any one time in the operating room. We start out with 4 teams, 1 on each recipient arm, 1 team on each recipient arm, 1 team on each of the back tables, so that's 12 surgeons operating at the same time. Then when the transplanted arm is prepared, John, we move it to 2 teams working side by side when we attach the arm to the body. So it's a huge team effort, 10 to 12 attending surgeons, but 30 to 40 people throughout the course of the 10 to 12-hour operation that are involved, all orchestrated and rehearsed.
I've seen some of the details on it. Simply the orchestration of the event is a feat in and of itself. Let's have one question here, and then over on the other side.
Surgical simulators to practice your technique.
Well, again, I mentioned this entity, one of the crown jewels in, I think, our educational paradigm now, called the Penn Human Tissue Lab. And it's a 2,000 square foot laboratory that I established with the support of Ralph Muller and Arthur Rubenstein when I came. That was the only thing I asked for, not a salary, not more faculty. I had to have my cadaver lab. And the lab has fresh cadaveric material. It's an operating room environment, and we literally have fresh tissue that we dissect and rehearse. I mean, we've used CAT-CAM modeling for models that we use to predict, for example, Zion's hand size that I carried with me to the procurement of the donor. It matched within a few millimeters. So we're using all the technology, pre-op, inter-op. We don't do computer modeling other than, it's a very good question, we use cutting guides for the osteotomies. We actually have computer-generated cutting guides based on the based on the recipient's forearm size and the residual bones in the forearm. We have these guides made, we slap the guides on, put 2 pins in, make the cuts perfectly on the donor and recipient. That cuts about an hour and a half out of the surgical time, and of course ischemia to the transplanted part is important, referable to neural regeneration. The longer the muscle goes devascularized, the less propensity there is for neural ingrowth to create functional movement in the transplant. So we do a lot of simulation and use simulators and the advanced technology for preparation, yes.
And I know in the cardiovascular field, more and more use of the 3D printers to mock up parts of the operation to practice ahead of time.
Exactly what we use.
Yeah. Dr. Potts?
There was a remarkable presentation by all of you. It's wonderful to be back here and listen to it all. And I don't know whether I'd ask any further questions, but I would like to zero in on the Alzheimer's story. As one gets older, one has to be thinking about that all the time, particularly. Do you believe that interrupting the tau transmission might change the course of the disease? And what do you think about the antibody therapies that we've tried? They haven't always worked yet, but they haven't done much.
Yes, I think that the antibody therapy, which has started already, it's very promising just because of the concept that the pathological tau has to get out of the cell before they can be picked up. So there's an opportunity for the antibody to catch it as it's transit from one cell to another. Yes.
Virginia, do you want to comment on the ideas around immunotherapy?
And this is the immunotherapy idea.
But not just an antibody, but targeted with a T cell or a...
Oh, you mean immune therapy? Yes. We actually started some of that work in trying to get T cell or B cells and or macrophages to express antibody, and so that they can go to the brain and then mop up and the pathology. We have this effort now initiated with someone, with Laura Johnson and Saw Gills in the Carl June's immunotherapy CAR T cells group. So this is really way into the future, that if we can trick immune cells to make the antibody so that they can go to the brain and then remove the pathology, and that would be the future.
Yes.
I didn't think I was old until I came back to the reunion. I guess I have to think about Alzheimer's as well. What about tau formation itself? How does that start?
Well, tau is a protein, and it's really quite abundant, that's required to stabilize these microtubules that are the transport system and in neurons. And so because neurons and with axon that can go so far from the brain to your big toe and then back to your big toe, so it requires tau to stabilize these microtubules. And because there's so much of it, and and also it's very stable, and we don't know what initiate the tau falling off of the microtubule and increasing in concentration in the cytoplasm so that they would promote aggregation.
Are there any animal models for this in the sense of dealing with sepsis? I remember, the name of the drug that came out. They were using it for a while. It was immunotherapy, and it just went away. And it seems we block one thing, and there's no single answer. And so I'm wondering in terms of if manipulating the biology, the microbiology in the cervix is going to be enough, or if there's ways that you can show in animal models that in fact is the deal.
So just so it may be hard to hear, the question is about really the microbiome of the cervix and whether manipulating that is going to be sufficient, and are there good animal models that could be used to test that? Somebody I know has made some of the best animal models used around the world, I think.
So you ask a great question, and I didn't pay him to ask it. We have developed animal models for the last 15 years. The problem is the first animal model I developed was exactly why Scott said. We were treating women, it sounds so loud, treating women with magnesium sulfate, right, to stop contractions. Magnesium stops cellular respiration in about every cell of your body, so you can stop uterine contractions as long as you stop her from breathing, so it kind of defeats the point. So I, when seeing this as a fellow, was saying, this makes no sense. So based on that paradigm I showed you guys, we initially made a mouse model to mimic that. We put LPS, a component of the cell wall of gram-negative bacteria, into the uterus, and we can recapitulate preterm birth 100% of the time without the mom getting sick or anything. But that really followed the old paradigm. So in the last few years, understanding how we've changed, we actually now have taken our good and our bad bugs that we found in our human cohort, so bench to bedside, bedside back to bench, and we now have mouse models under the funding of the March Dimes Prematurity Research Center, where we actually have created, using G. vaginalis, mobiluncus, we are creating actually a vaginal human microbiome in the mouse, and we have found that there is obviously some mechanism that these induce cervical remodeling. They don't cause preterm birth. Every species aside from us seems to not have preterm birth. That's another question we should ask. But we are able to recapitulate the cervical remodeling, and we're using those to test therapy at the moment.
So this starts to get at the question of causality. That's so important. If I just can relate sort of related story, everybody's aware of the excitement around understanding more about the microbiome. It does seem to affect absolutely everything in surprising ways. Yesterday, excuse me, in the New York Times, Gynecology wrote a very nice article you may have seen about a Nature or Science paper, I can't remember, of Mark Kahn's, researcher here in the Cardiovascular Institute, that just came out. Really remarkable whodunit sort of story about cerebral cavernous malformations that are a fairly rare cause of aneurysmal-like disorders in the brain. Mark has worked out over the years the genes that cause this inherited genetic abnormalities and a pathway, all 3 genes that have been found in humans work in a molecular pathway that he's helped to decipher. But in the middle of his studies, some annoying chief scientific officer moved his laboratory from the BRB building to Smilo, right here. And that meant moving his mice to a different animal room. And everything went to hell. The mice with the genetic mutations that he'd engineered to recapitulate the human disease stopped getting cerebral cavernous malformations, at least in 2 of the 3 mutations. And Mark never takes unexpected results lightly. He digs in like nobody I know to find out what's going on. And the upshot is that the microbiome in the 2 animal facilities is different. And the only time these mice, when they moved to the new cleaner facility, would get malformations was when they happened to get an infection, usually from injecting them with drugs that they were testing, which led to an abscess or something like that. And he noticed that only when they had an infection would they get the aneurysm, and tracked it down to gram-negative bugs populating the gut that would occasionally get in the bloodstream, which was required in addition to the genetic abnormality to cause aneurysms in the brain. And he did fecal transplants from one to the other. He recapitulated the results with LPS. I mean, he went to extreme. But it's a remarkable demonstration of what types of even functional malformations might be caused by changes in our microbiome. So it was fun to see it picked up in the press, and actually I thought a very good article by Gynecology describing the results. I think our time is, are we done at noon or 12:15? Noon? I think maybe we should stop it here, thank our panelists, and thank all of you for joining us.